right, so let's talk a bit more about phylum Mollusca, and let's learn a little bit on how Mollusca is, what organisms it contains and things like that. So the phylum Mollusca is the second largest phylum, and it's second largest only after in the marines. So it's over 100,000 species, and that's species that we've already found and cataloged. There could be more, right? Because we know less about our own ocean than we do the surface of Mars. So could be thousands more, who knows? But there's over 100,000 named species in Mollusca. So Mollusca is characterized by shells, all right? So they are all invertebrates, soft-bodied. They have a mantle that forms a shell. So that's important to know. Mollusks have shells somehow, some way. All right, so they have a muscular foot for movement. Now, it's not like a foot like you would think of your feet, but it's a type of foot. We're going to learn what that is. It's just it's a muscle, but it's sometimes modified into tentacles. So one-way digestive tract, which means that it's mouth all the way through to an anus, which is important because up until now, none of the organisms that we've talked about had actually had an anus, which sounds ridiculous. They have cephalization, which means they have a head region, and have a distinct brain area, which is, again, different than all the cnidarians that we talked about because they don't have that. They have a scraping mouth-like structure called the radula. And remember when the Gulf Specimen Lab came over, they were talking about how it scrapes a hole into other mollusks? That's what he's using, is a radula. All right, and they have free-swimming larvae. So all of those characterize what makes them a mollusk. So you have to understand what makes a mollusk a mollusk. So all three of these things are mollusca. So you have gastropods, which are the typical snail-like organisms. You have bivalves, bi meaning two, valva. So these are like the clams and things, anything that has two shells, right, that are held together. And then cephalopods, which are really unique. These are your octopus, obviously, but it's also like cuttlefish, things like that. Squid, they're cephalopods. So there's a lot of different things. So the first one that we're going to talk about, today's lecture is all about bivalves. So what are bivalves? So bivalves are obviously clams, right? These are scallops. They have two shells that are held together on one end with a ligament. So if you ever found this, if you went to a beach and you see these shells stuck together still, that's how they live their life, guys. They live their life together. And they're kept closed with a really strong abductor muscle. And they're really hard to open. We're going to do a dissection with a clam, so you're going to see these little suckers are hard to open. They have very strong muscles. All right, so that muscle is actually located on each side here. So you have the hinge, you have the two really strong abductor muscles. So you have an anterior and a posterior, remember, towards the head, away from the head, right? And this is an open shell already, right? Obviously, the second shell has been taken off, so you can see this. So without it, you can't do that. All right, so this is how they should look. If you ever got mussels? Anybody here eats mussels or clams? They're really good. If you haven't tried them, they taste better than they look, I promise. All right, well, they look like this, and you have to like really force them to get them open, like really force. Now, if you cook them, they pop open, right? Because they're dead. But if they're open and they're not supposed to be, don't eat them, okay? All right, that's because they're rotten. They're not good. But these right here, so this is the muscle. Anybody here go scalloping? Because we're famous for scalloping here, right? All right, they're really good. You like sea scallops? They're like circular. That's that muscle. That's all it is. Everything else we kind of throw away with the scallop, but we do keep that muscle because it's a lot larger in a scallop. All right, so this muscle here, that's the posterior abductor muscle, and the anterior one's on this side. So they have these two really big literal muscles that hold them together. All right, so bivalva have shells. These are shelled organisms, just like we talked about. So they're going to be affected by, global warming. They're going to be affected by ocean acidification because they are calcium fixers. So they're calcium carbonate shells. So it's made of that white powdery substance that we got to fall out of solution when we were doing the blowing. Remember on that lab? All right, so this, the mantle itself. So these organisms build their own shells. They come from them. All right, so they make them. And the membrane itself, there's a membrane on the inside. If anybody's ever eaten oysters, you know what membrane I'm talking about, the one that lines the shell. That actually makes the calcium that makes the outside, okay? Well, it actually fixes, I should say, instead of makes it. But it produces the shell itself. So these organisms are making their own shells. Uh-huh.
No.
I'm sorry? I don't know what the dealio is with it. I'll fix it in a minute. All right, so the mantle is right up against the shell, guys. It lays on it. It's just a film. It looks like a membrane, and it lays on that shell. And this is the mantle in a real living one. So the mantle lays on the shell. It's the furthest one. It's the closest to the bottom, right? Or the top if you're the other way. Looks like a fried egg to me. Doesn't it kind of? All right, so the shells are shut really tightly with just a small gap. And they have a little siphon that comes out. They have a siphon for water, okay? And it's like a tube or a straw, but it's made from them. And they tend to live buried in the sand, and they stick that little siphon up. If you've ever been to our beaches here on the Atlantic side, you'll see these little holes in the dirt. As the wave comes up and goes back, you'll see these little holes, and they'll tend to spit up, and they look like they're bubbling sand. That's the siphons from all of the bivalves that are in the dirt. There's a lot of them in the beach sand. But that's how they live. They live down below. They stick their siphons up. And they can be really large. Like in a gooey duck, they can be feet long, like long, long, long. If they're small ones, they'll be smaller. It just depends on the size of the bivalve. But they use this siphon to do that. All right, so when you look at them, their siphon is actually, it doesn't really show it very well, but it's like the end of a tube, okay? Now, they can suck this back in because it's flexible, but it's just the end of a tube here. So it's trying to show you that it's an arched way. And then in a living one, this is his foot. If you've ever been lucky enough to go out to the beach, you can see these guys trying to dig in the sand, and they use that muscular foot to do so. It looks more like a tongue than a foot to me. Doesn't it kind of? It's kind of sticks out. But that's what it's called. It's called a muscular foot. And they use that, and they just kind of stick it in, and then they go all the way down into the dirt. They burrow themselves in there. Come on. So here's a video. Oops. Let me go back. You all right? Don't fall. No. Is it playing? No. It's not wanting to play. Let me fix it. Oh, I know why. All right, so in the video, what we're going to see is we'll see them bury themselves in the ground. It won't play when it's on Google Slides. It has to be on this. All right, here he goes. Watch the video. Watch what he does. So he uses this foot, and it sticks out. So he uses this foot, and it comes out, and he's going to bury himself using that muscular foot. It looks like a tongue, I know, but he's going to, you see the little fish in the background. That's what that fuzzy thing is. But watch what he does. He'll bury himself rather quickly. See his siphons over here on this side? See how they're sticking out? They're frilled. They're the ones that he's using to pump water in and out of his mantle cavity so he can breathe. So he's going to put those to the top. See how he just kind of wiggles? And he can get into the dirt, and it protects him. See his siphon's a lot better now on this side. See the siphons, how they're holes? You can see the opening and closing. One pumps in, one pumps out. And then he'll just put those to the top. And that's how they live, buried in the sand. They can be a couple of feet down. They could be meters down or inches. It just depends on the species itself. And they can stick those siphons out further. They can extend them. Some species can extend them a very long ways. And that's how he protects himself, right? So he's fully buried so that nobody can eat him. That's the point of it. He's trying to protect himself from being eaten. All right, so they're filter feeders, guys. So what does that mean? What's filter feeding? Come on, we talked about it when the Gold Specimen Lab was here. What is filter feeding? Nobody has a clue. Are they eating filters? No. What are they doing? All right, tell me.
I don't know.
They clean the water, yes. Of what?
The area around them?
Of what, sorry? Kind of, not really. They're cleaning stuff out of the water. What is in the water? We did a lab on this. What's in there? Well, yeah, unfortunately.
The low part of the ocean.
What are they eating?
Plankton.
Plankton. Very good. They're eating krill, if that's a little big, but maybe small little larvae krill. Zooplankton, or zooplankton rather, right? And then they're also eating phytoplankton. They're eating anything basically in that water. They're pumping it in with the siphon. They're filtering it out and pumping the water that's empty back out, right? They're just constantly doing that. Now, Elizabeth did bring up a good point. She said, what about the toxins, right? Yes, they do that. You ever heard that if you're pregnant, you shouldn't be eating certain foods, especially seafoods? A lot of that is shellfish. And one is because, if you have an allergic reaction, it could hurt the baby. But the other thing is because these are filter feeders. They're at the bottom of the food chain, right? Or somewhat the bottom. They're almost. So these guys are primary consumers. They are cleaning out the water, and it is possible for them to pick up some diseases that have been in the water. So if it's a very polluted water, they're going to be picking those up. So it could be that if you eat them, you'll have a large buildup of these pollutants in you as well. So that's one of the reasons why. They're also the cleaners of our ocean. If we lose these guys, our seawater immediately gets nasty, very nasty. They keep the water crystal clear. That's what you want. You want these bivalves because they are literally the scrubbers of the ocean. They're keeping it clean. They cleanse large amounts of water. They indicate water quality. If you have a large population of bivalva, your water quality is probably going to be a lot better in that area than places that have lost their bivalves. So during filter feeding, water, organic debris, plankton, whatever, passes in through its siphons. See the siphons in the sand here in this photo? These are larger. These are really big. And you can see the water coming in, and that's what they do, is they suck this water in, and everything in it, they're basically going to filter out. So they get stuck on the mucosal surface that lines the gills and the mantle. And if you've ever ate raw shellfish, you know this, they're kind of gooey, right? You ever opened up an oyster and ate it raw?
Yeah.
They're slimy. That slime is what helps them filter feed. But the oxygen is absorbed. They actually have gills. It's interesting, a different type of gills, but they do have them. Hello? Mm-hmm. Okay, thank you. Tyrese, can you go see Miss Ramona real quick?
Oh, no.
All right, so these guys do have gills. All right, so here's a little video. Look at the water quality. This side tank over here does not have any oysters. This tank does. And they put it in a time lapse, and look what happens over time. Look at that water. What's it doing? What's the water doing? It's cleaning it, right? It's getting a lot more clear. Look how much difference. This is just without them, same water. And then with them, look what happens. It gets a lot better, doesn't it? You can prove that they really keep the ocean clean, and they'll make that water crystal clear. So it starts out like this, and then you're going to see in 24 hours time, they clean it to almost perfect. Mm-hmm. That's what the water looked like at the beginning. So these guys really, really clean the water in the area around them. See, that's what it looked like beginning. All right, and they do that by using their siphons to pump water through their mantle and out through the gills. So the gills keep it, give them oxygen from that water, and the mantle helps provide them with food. All right, so the bivalves, oops, I hit the wrong button. All right, so the age. We can determine the age of the shells by looking at the rings, and we're going to do this next class. So each ring represents a year of growth. You see on this shell, you have the first ring here, the second ring, the third ring, and then he's building on to the fourth, right? So this shell is 3-point-something years old. You can date these shells based off of these rings. So each ring is a year. So we're going to give you shells to read, and we're going to figure out the age of the shells that I give you, because everybody has another one. So make sure that you know that each ring, each dark line, basically, and it has to be a continuous dark line, because some of them are darker than others, will tell you how old the shells are. All right, so this class Bivalva, in the phylum Mollusca. Make sure you know that. Phylum is larger. One class that we're learning about is Bivalva. That's the first one we're talking about. We'll get to the rest of them in a couple of days. So they have an open circulatory system, which means it's just kind of open. The blood is bathing and not necessarily in all of the same as ours. It's not the same. It has an open system. All right, so it means the heart pumps blood into vessels. The vessels open up into just open chambers where where there's like blood bathing all the organs. They don't really have integration like we do. It's a little bit more primitive. We have a closed circulatory system where everything is kept in a vein and an artery. Theirs, they open up and get these little sacs around their organs, which are just bathed in blood. But they just do the same thing, though. They just move blood around the body. Bivalves move. They do move. They have that foot. I showed you the video earlier where you could see them moving. And they can propel themselves, kind of. Okay, so the way they propel themselves, you got two shells, right? They just kind of go like that. It's nothing special. But the scallop, the sea scallop, is actually the fastest. And if you ever go to to Keaton Beach and you're swimming in there and you're looking at scallops, they can actually like move away from you. They're not like the best, but they can move, you know. All right, so this is a video of a scallop swimming. So watch him. See how he uses it? He's going like this, and he'll swim away. See, he can move more than you think. It's kind of funny to me. I don't know why. But they can move. They can swim away. They're not the best, but they can do it. All right, so we eat them, obviously, right? What is this? A pearl. You know that, right? And you know that they come from oysters, not our oysters. You can get pearls, but they're rare. Usually the ones that we get, like the pearl necklaces and things, those come from Indonesia or Polynesia. All right, so it happens whenever something irritates it. So like farmed pearls, we can make them make a pearl. All we have to do is like put something like an irritant in there. And if it's irritating to them, all they can do is that mantle will secrete some calcium, and it's just going to cover it and cover it and cover it and cover it. And that's when you wind up with the pearl. So it's food or sand, something gets lodged in there that they can't expel. And they just cover it with layers of shell material. And it's really pretty. And it just gets formed around and around and around it. They're not all perfect circles. Some of them are kind of wobbly, but they're really pretty. This is a scallop flying away. Watch him swim. So if the predator gets near, see how he's using his foot? So this sea star actually eats these, so he can go away from them. You wouldn't figure, but he can. He can get away and actually get away faster than the sea star. So they can move. So you will be required to know all the parts of it, because next week we are going to have our first dissection, and it's going to be the clam. Because one, it's easier and they're really simple. All right, so you will need to know all the parts of the clam, especially before we go into our dissection, because if I ask you to cut through the anterior abductor muscle, you need to know which one that is, okay? So the anterior is always closer to the hinge. That's how you know that. You might want to write yourself a little note. The anterior is always closest to the hinge. And the posterior is usually larger, and it's further away. So if you can't write these in on your notes, by all means, just number them and write them out to the side so I can see it. I know it's small. We're just trying to save paper. So just put numbers on them and then just number them out to the side so we know which ones they are. All right, we're going to talk about cephalopods after our lab, okay? So we're going to learn only about bivalves this week and next week, and then we're going to move on to cephalopods for 2 weeks. And then gastropods are really small, so we only have a week for those. All right, so we've got about 4 weeks left, and we're going to do the clam dissection first. Then we're going to do a squid dissection, and hopefully we can get to the larger. We have one more that's after that for the arthropods. We have a crayfish. Hopefully we can get to it. I'm not sure if we'll make it in time. Depends on the year, right? All right, but we have a bunch of big things coming up. Remember, during dissections, we have to have closed-toed shoes, jeans, because they're actually cut resistant. They're actually parts of your laboratory stuff. You need to remember all parts of the dissection kit and all of your safety information with those. So if you need to go back and review, it's still posted. Sir?
What we doing?
We're going to do the clam first, and then we'll do the squid, not the octopus.
We do, didn't we?
They're expensive.
We do,